Thank you. Good morning, everyone. Today we have the pleasure of welcoming Dr. Arazi, who will tell us about myeloid cells in lupus nephritis. Dr. Arazi did his PhD at Bar-Ilan University and then did a postdoc at the Broad Institute. Currently, he's an assistant professor at the Feinstein Institutes, and he has done instrumental work in the Accelerating Medicines Partnership, which I think most people here are familiar with that. It's a NIH initiative with multi-institutions and with the goal to better understand and characterize lupus and other autoimmune diseases. And Dr. Arazi was involved in phase 1 and also now in phase 2, he's leading the effort in better studying lupus nephritis. So we look forward to a very interesting talk and discussion.
Thanks, Ryan, for this introduction and also for inviting me to speak here today. Can you hear me fine?
Yes, we can hear you well.
Okay, let me share my screen. Work. And we should be able to see the presentation. No, can you see it?
Yes, now it looks good.
Okay, so the work I'll be describing today, it's the product of several years of like Ryan said, a very large project. It's actually pretty close to being published specifically, or at least submitted for publication. Specifically, the part I'll show today, I'm hoping kind of ambitiously to submit it today. There are a few other papers coming from this, which I'll kind of just briefly mention. One of them, by the way, is looking at urine cells, which I won't be speaking about today. And this one was led to large extent by Ryan himself when he was working with me a few years ago. But we'll keep this for another time. So the work that I'll describe here really started with one general question, which is how to improve the drug development. And as I'm sure you know, currently this is a very expensive and inefficient process. We spend billions on these drugs, and the large majority gets to phase 3 and then fails. So we definitely need to do better than that. And one hypothesis is that the reason why this doesn't work that well is that we rely too much on animal models, that instead we should study more directly human disease. Now, of course, this has its difficulties because you cannot manipulate human subjects the way you do with mouse models, right? So you need to somehow use a different approach. And one way to think about it is that you would like to maximize the amount of information that you get from a limited set of samples that you can obtain from human patients, especially tissue. Now, we cannot do any point perturbations with human patients, but we can still utilize the natural variability of human populations and try to use that to infer at least something about disease mechanisms. It's never going to be a complete picture of like we have in animal models, but it can feed back to more specific and more relevant animal models. And that's kind of the path we're taking. Now, as Ryan mentioned, this work was done as part of the Accelerating Medicines Partnership, the AMP, which is a joint initiative of the NIH, a number of pharmaceutical companies and nonprofit organizations. And this started about 12 years ago, I think. And this initiative had the general goal to improve the way we develop drugs to make this more efficient. And again, with the notion that I just mentioned that we need better usage of profiling technologies and new technologies that would allow us to gain maximum information from human samples. Now, a few diseases were selected as part of this initiative to demonstrate this approach, and one of them was lupus nephritis. In lupus nephritis, I probably don't need to give you much introduction of this. There is a need to get better therapy. So it's a relatively common manifestation of lupus. About half of the patients eventually get it. And even though we have new treatments available in the last few years, response rate is not great. So in most studies, you don't get much more than 50%. And many of the patients that do respond eventually relapse. So what we do need is more specific and more effective drugs. And we also need to have some way to have a personalized application of medicine. So to know which drug to use, what subtype of disease, and what stage of disease, which eventually means that we need a better understanding of the mechanisms of disease, especially for lupus nephritis in the kidney. Now, currently the way clinicians, again, I don't think you need this information from me, but the way we characterize heterogeneity in lupus nephritis is mostly through kidney histology. And we have the ISM class, and the general distinction is between proliferative and membranous disease. And then there are two composite indexes, the NIH activity index, which generally measures the degree of active inflammation in the kidney, and the chronicity index that measures the degree of irreversible tissue damage. And what we do in this work, we're going to utilize the natural heterogeneity of lupus nephritis and look for correlations at the cellular and molecular levels with these histopathological features as they are measured by the ISM class activity and chronicity. In addition, we're going to utilize the fact that, by definition, irreversible tissue damage just accumulates over time. So in general, the chronicity index should increase over time. So if you can find correlations with the chronicity index, this allows us to start thinking about time. So think about disease processes with the idea that if something is, let's say, positive correlated with chronicity, it should increase with time. So that even though we have just one single time point per patient, because getting more than one biopsy is almost impossible, we can still use this natural heterogeneity and the idea that the CI just increases, the chronicity just increases with time, and translate these results to some picture of disease process. Again, it's not a definite description, and it would be great if we had some way to follow patients over time, the kidney state. And we do develop some ways, I should say, through urine. But in this study, and what I'll show you today, we're using just the kidney samples themselves. Okay, so for specifically lupus, the AMP was focusing on lupus nephritis. This was a large-scale multi-center study. We ended up profiling kidney samples from 156 lupus nephritis patients, 30 healthy controls. And we looked at a bunch of different samples. For kidney, we had single cell and single nucleus RNA-seq, and also histological information. We did single cell RNA-seq on urine, also proteomics, and there's also profiling of blood. In today's talk, I'll just focus on the single cell RNA-seq of kidney biopsies. Now, the AMP, again, was a pretty large initiative, and as you can see, many people were involved in it. I'll focus on a few, as I mentioned during this talk. As Ryan said, I started my own work in the AMP as part of my postdoctoral training at the Broad Institute, working with Neha Cohen. And then a few years ago, I moved to the Feinstein Institute to have my own group there. And I collaborate mostly with Betty Diamond and Ann Davidson, which played central roles in the AMP and still are playing central role in the AMP project. So this is still an ongoing project with various stages. So what we did here was collect kidney biopsies, and these biopsies, what we found is the best approach that decreases batch effects is to take the whole piece of tissue and just freeze it in the clinical site immediately after processing. And then these frozen samples were sent to a central processing site, and at the latest stage, with batches of several biopsies at once, the samples were thawed, enzymatically dissociated, then loaded into 10X. We used a protocol called DASH to remove excess mitochondrial reads, just to improve the efficiency of sequencing. And following that was the analysis. And this protocol was developed and later implemented by Tom Eisenhower, who is an exceptional scientist working at the Broad Institute. In terms of inclusion and exclusion criteria, you can see them here. we are aiming at patients with relatively progressed disease, so class 3 and 4, and also pure membranous. The patients at the time they were recruited were flaring, so active kidney disease. And again, I mentioned we had 156 patients. Of these, one patient did not have leukocytes in her kidney sample, but the rest did. We have a nice coverage of race, ethnicity, and classes. So we're well set to characterize heterogeneity here. And we are trying to answer a few main questions. First, which immune cells can you find in the kidneys of lupus nephritis patients? Second, what is the relation between the relative frequencies of the cell subsets and the histology as represented by the ISN class, chronicity and activity index? We are also looking within cells at the molecular pathways that are active in them and the relation to these features. We are asking where the immune cells are in the kidney, and can we learn anything about where differentiation takes place? And finally, for this talk, we're also looking into interactions between immune cells and tissue cells and see how they may be driving disease. Now, I'll be focusing mostly on myeloid cells here. I will mention some of the other immune cells, but it's going to take too much time to speak of more than one cell type, so it's going to be mostly myeloid cells. So starting with the first question, after cleaning the data and removing low quality cells, we ended up with around 74,000 immune cells. About 1/3 of these were myeloid cells. T cells and NK cells were a bit less than half of the cells. We have a separate work looking at these. It's actually close to being accepted for publication, I think. It's a collaboration with Joe Kraft from Yale. What I'll just mention briefly here is that we could see here both CD4 and CD8 T cells. So we had various populations of memory CD4 T cells. And in the CD8 side, we had granzyme K CD8 T cells, tissue resident cells, and CTLs, and also plenty of NK cells. And we also see here B cells and plasma cells in lower quantities. I won't be speaking about this today. Now, within the myeloid cells, we had about 24,000 high-quality cells. And you can identify several subsets. We could see both classical and non-classical monocytes, and two populations of tissue-resident macrophages, several subsets of dendritic cells, and then two sets of clusters that were more disease-specific. One, what we call differentiated monocytes. So if you compare these cells to reference data set, it looks like these are mostly monocytes. We don't really see here tissue-resident cells. But they tend to be disease-specific and have various states within them. And a large set of what we call disease-associated macrophages, so DMACs. And these cells, if you look at where the cells from the healthy individuals were, so LD is that's living donor, this is the healthy controls here. So the cells from these living donors were mostly on the monocyte side, and the tissue-resident cells, some DC2s, but almost no DMACs, so disease-associated macrophages, almost no differentiated monocytes. So these sets of clusters were more really related to lupus itself. If you look at the cell subsets that do appear in the healthies, so again, monocytes, the two populations of tissue-resident cells, and DC2s, and you just look at the interferon response, as expected, we see an upregulation in the lupus patients, so that's not surprising, but it's something to look at. But then when you try to understand what's the different signatures that distinguish between the different subsets of DMACs, for example, we see various different flavors here. It's various transcriptional programs. So for example, some DMACs, like the ones I circled here, have a relatively low interferon response. So the dashed line in all of these is the average across myeloid cells in lupus patients. So these would be less than the average. Other subsets have high expression of genes associated with phagocytosis. So you can kind of at least hypothesize about their function as well. And most of these subsets had a high expression of signatures that's associated with tissue injury. but again, not all DMARDs. So we can kind of use this combination of transcriptional programs to start thinking about the possible differences between the subsets. Okay, going to the second question, what is the relation between the relative frequencies of these different myeloid cells or other immune cells and the ISM class chronicity and activity indices? We first asked if we can identify distinct subsets of patients based on the composition of immune cells in the kidneys. So could we actually see clear and separate subsets of disease? And the answer is no. What we did see instead is a continuous and gradual change in terms of immune cell composition across our cohort. So we use the continuous approach named the PCA to characterize this variability. This allows us to identify the main sources of variability in the data and also to see how they correlate with the chronicity activity in ISM class. And when we did this, what we found is that the first principal component, which explains about 1/3 of the total variance, roughly represents the balance between selected lymphoid and myeloid cells. So on the one side, we have the differentiated macrophages with DMACs and monocytes, also differentiated monocytes. And against that, we have the B cells, granzyme K CD8 T cells, and different subsets of memory CD4 T cells. So what we found is that this PC is correlated with the chronicity index, such that patients that have higher chronicity tend to have a higher lymphoid to myeloid ratio. So they have more of these B cells, granzyme K CD8 T cells, and CD4 memory T cells, and less of the monocytes and macrophages. And this was true both for proliferative disease and for pure membranous if you analyze them separately. Now, as I mentioned earlier, the chronicity index, since it represents irreversible tissue damage, should just increase during over the course of disease. So we can use this information and think about how disease changes over the course of disease as it progresses. And what this tells us that there should be a gradual shift from a response that dominated by macrophages and monocytes. And I'm not mentioning this, but when we look at T cells, we also see that the CTLs are mostly expanded with low chronicity, and then they go down with high chronicity. So we start with something which is more cytotoxic and involves also monocytes and macrophages, and then move to something which is more related to the CD4 T cells and B cells, so perhaps also more antibody drive. Also antibody driven. Like I said, we see also an increase in granzyme K CD8 T cells, but based on our analysis, this is mostly because there's less differentiation in the CTLs. The granzyme K CD8 T cells represent an intermediate stage of differentiation, an early stage of differentiation in CD8 T cells. And once you kind of stop the differentiation in the CTLs, you have an accumulation of these cells. And indeed, when you look at the PC1, or also the ratio of these lymphocytes and monocytes, and the relation with the age of the patients at the time of biopsy, we see a significant correlation. So again, older patients, which you'd assume have more progressed disease or had more time to be sick, they tend to have more lymphocytes and less monocytes in the kidneys. Now, again, this is not a direct proof, of course, and it would be great if we could follow patients over time. We actually ended up getting some repeat biopsies. It's a small subset, so it's not a full description, but right now we're looking at them to see if they can validate the results. See here, and it does look, yes, I mean, it does look like the right kind of immune subsets go up with the repeat biopsies, especially when these patients are not responding to treatment. Okay, now the second principal component, which together with the first one explain more than half of the total variance, seem to be representing the balance between differentiated macrophages and circulating monocytes. So you can think about it as representing the extent of DMAX differentiation. And what we found is that this one is correlated with the activity index. So active inflammation means more differentiation in towards the macrophages and less of the monocytes. But this was true only for the proliferative patients. In pure membranous patients, we don't see this relation. So this led us to decide to analyze the proliferative and mixed patients separately from the pure membranous. Mixed disease is just very similar to proliferative disease, so we just group them together. and so I'm similar in terms of the molecular features and cellular composition of immune cells. So what we did for each ISN class, the proliferative and the pure membranous, is define two sets of patients, one that had low chronicity and one that had high chronicity. And this is based on a specific choice of thresholds, but what we show in our work is that the results that we get are not dependent strongly on this choice. So we can vary these thresholds and we get basically the same results. But once we define these groups, we can think again about one as representing early disease, the one with the low chronicity, and the one with the high chronicity as representing more progressive disease. And then we can compare the early disease to the control. So that would tell us something about what's going on early in disease, and a comparison of the patients with high chronicity versus the patients with low chronicity will tell us something about what changes as the disease progresses. Okay, so doing this analysis, we started looking at changes in cluster frequencies, and we use an approach called the covariant neighborhood analysis, which looks at how the frequencies of different subsets of cells correlate with some clinical variables. So when we use it here, what we found is that when you compare the early disease to the healthy controls we see in the lupus patients, in expansions of the DMAX, the disease-associated macrophages, and we actually see lower relative frequencies of monocytes and tissue-resident cells, which, again, is consistent with the idea of differentiation. So active inflammation means differentiation towards these macrophages. And then later in disease, what we see is a contraction of these disease-associated macrophages, at least some of them, and a relative expansion of the dendritic cells. Now, if you think about the result I showed you earlier when we're looking at all immune cells, so this is just looking within the myeloid cells compartment. But when we're looking at all immune cells, I just told you that we think there's a gradual shift towards something that's more centered around CD4 T cells. And this is consistent with the expansion of dendritic cells, right? Thinking about that they may have a role in activating these CD4 T cells as APCs. Now, in a pure membranous disease, we don't really see that. So there's no expansion of the DMAX early on, or minimal expansion. And then later in disease, we do see in pure membranous an expansion of the dendritic cells. So this seems to be a general result. We next looked at the pathways that are active in each group of patients. And again, we're using the same patient groups as before. And what we did is per cluster run differential expression analysis, comparing these groups. So again, the early disease versus the healthy, and then the late versus early disease. And we interpret the results using gene set enrichment analysis, focusing on specific pathways that are relevant for the function of myeloid cells. And what we find first, and there's a lot of results. I'm going to hide some results from you in each step and just focus on one pathway at a time. So starting with the interferon response, what we see is that early in disease, as expected, there's an upregulation of the interferon response in lupus compared to healthies. And this is, for now, I'm showing you the proliferative disease. But then as the chronicity increases, there is a decrease in most clusters of the interferon response. So the interferon response seems to be a feature of the initial disease, not so much of the later disease. In pure membranous disease, we see something similar. Again, an increase of the interferon response early on. And then as the disease progresses, a decrease of the interferon response compared to the early stage. Now, if you compare the late disease to the healthy patients, what we found is that in pure membranous disease, the interferon response goes almost completely to normal levels. It's still higher in proliferative disease compared to healthy controls, but at least in the membranous case, it basically is just a feature of the early stage of disease. Another pathway we're looking at is associated with the processing and presentation of antigens. And what we found is that this is upregulated early in disease, but then later in disease, it still continues to go up. So unlike the interferon response, these cells become more capable of antigen presentation. And that's true but for the monocytes, for the DMAX, tissue-resident cell, dendritic cells, all of them go through this change. In pure membranous disease, we see something similar early on, but not so much of a difference later. And actually, in some clusters, this machinery goes down. And again, this change in APC functionality is consistent with the shift if the immune response toward one that's more centered around CD4 T cells. If you think about these myeloid cells as becoming better APCs, and that's again true not just for the dendritic cells which expand, also the DMACs, which, like I said, contract, they also change their function and becoming better APCs. Another pathway we're looking at is phagocytosis. And as you can see, it's in proliferative disease, it's early on and up in most clusters, and then it goes down as the chronicity decreases. In pure membranous, we see less of that. The tissue resident cells do have an upregulation of this gene, but not the monocytes or the dendritic cells, and not much of a change later. So again, thinking about the greater story here, if you think that there's a shift away from something which is more cytotoxic, like I said, the CTLs also go down later in disease, then you actually expect to see less of the phagocytosis because there are less dead cells around. I mean, that's one interpretation. And this can also explain why there's less of the interferon response. Now, you can think that these changes, specifically the decrease of interferon response, are just due to changes in medication. Maybe when the patients are more sick, they're treated more aggressively. But what we found is that when you take the treatment as a covariable here, as a covariate, you still get the same picture. So, for example, for patients treated mostly with MMF, mycophenolate, you still see a decrease in the interferon response, both in proliferative disease and in pure membranous disease. Same goes for high-dose steroids. So this by itself doesn't explain the changes we see in gene expression. Okay, so just to summarize what I've shown you so far, what we think is happening is that both proliferative and in pure membranous disease, when the chronicity increases, so as the disease progresses, we have a gradual shift. And I should say this is not a one and all shift. It's really a continuous shift from an immune response that's mostly led by macrophages, monocytes, and CTLs into one that's driven mostly by CD4 T cells and B cells. And we see this in the relative changes of immune cell subsets, both within all immune cells taken together or within the myeloid cells themselves. And we also see changes in the transcriptional programs. We have less of the interferon response and more of the antigen processing and presentation machinery. And only in proliferative disease, early disease is associated with expansion of these DMACs, which later go down as the disease progresses. And parallel to this, we see an early upregulation of phagocytosis genes, and then they go down as the disease progresses. Okay, so changing gears a bit, we're asking where in the kidneys are the subsets and where do we have this differentiation in DMACs? So to answer this, we used Xenium, so spatial transcriptomics. And as you know, this is still a pretty new technology and pretty expensive as well. So what you see here is a much smaller cohort, 6 lupus nephritis patients, 2 controls. But since we're mostly interested in the expansion of DMACs, we focused here on proliferative disease with low chronicity, so early disease, because this is, like I just said, this is where you see the expansion of these cells. You're not going to see them in pure membranous patients or later in disease. So even though we have a small cohort, this allowed us to maximize the amount of information we can get from it. We designed a gene panel that allowed us to capture all 24 subsets of myeloid cells that we see in the single-cell RNA-seq data. So it's really a myeloid-focused gene panel. And the work I'll be describing in the next few slides was led by Paul Hoover. He started as a postdoc at the Cohen Lab at the Broad Institute together with me, and now he started his own group at Brigham and Women's. So how do we know that this actually works? What we've done is, I'll walk you through this figure. We took the 24 subsets we have myeloid cells in the kidney, so that's what you see here on the left, And we identified sets of genes that allow us to distinguish between them. So we actually do this hierarchically. So first of all, distinguish between groups of subsets and then going down to more specific comparisons. And then you take the xenium data, you take the myeloid cells there, which we identify through clustering, and then you map them to the kidney data. So this is how you label them with the same labels that we have in the single cell RNA-seq data. And when you look at the signatures that you get in these mapped clusters, they are very similar to what you get in the original clusters in the single cell RNA-seq data. So we can reproduce the signatures that distinguish between the cells in the kidney in the single cell RNA-seq data with the spatial transcriptomics data. So this tells us that this approach is working well. Okay, so asking first, where do we see these different subsets? And the first observation, if you just look at the fraction of cells per cluster within different compartments of the kidney, so the glomeruli, the border of the glomeruli, the periglomerular information, the inflamed TI or the uninflamed parts of the TI. What we see is that most cells are basically everywhere. The largest fractions in almost all clusters are where you don't have any inflammation. But this is really just a game of numbers. It's much a larger area of the kidney that's not inflamed, and therefore you just see more cells there. But when you start normalizing this and thinking about probabilities and looking preferential special patterns for the specific cells, we do see enrichment for specific compartments. So I'll walk you through this figure in the next few slides. First of all, some subsets of cells, mostly differentiate monocytes, and some subsets of DMACs, disease-associated macrophages, seem to be preferentially localized inside the glomeruli. And I'm showing you, I'll show one slide here as an example, each time highlighting another group of cells. So you see, these are different subsets of DMACs, and they tend to be inside the glomeruli. Another set of differentiated monocytes and DMACs seems to be just next to the borders of glomeruli. So it's very difficult to know exactly what the border is of where the glomerulus ends and where it begins. So we define this border area that has a width of 30 microns, although we can play with this, it doesn't change the results, the definition of this width. And what you see is that these cells concentrate on the border of the glomeruli or around them in the periglomerular inflammation. And I'm showing you a few examples of such cells here. Now, dendritic cells seem to like mostly the inflamed parts, inflamed aggregates of the tubular institution, or they're also around the glomeruli. And again, you can see here some examples of that. And then the tissue resident cells, the undifferentiated tissue resident cells, are all over the tissue, but they seem to have a preference to the uninflamed parts of the TI. Now, when we're comparing our single-cell RNA-seq data of the kidney to reference data sets, both of healthy kidneys and healthy blood, what is suggested is that the DMACs are really a mixture of cells coming from the tissue-resident cells and monocytes. So this led us to hypothesize that there's something called convergent differentiation here. That is that the monocytes that infiltrate the kidney and the tissue-resident macrophages acquire over time similar transcriptional states as part of disease. So we wanted to study this a bit better. And first of all, we checked the plausibility of this hypothesis. And we started with a trajectory analysis of the single-cell RNA-seq data. We chose a specific tool called PAGA, which in general performs well when you have a benchmark test comparing different trajectory analysis tools. And it also allows a very general structure of transition. So it allows several starting points of trajectories, which is what we need in our case. We have 4 different possible origins of cells, 2 populations of monocytes and 2 populations of tissue-resident macrophages. Now, what PAGA does is assigns a confidence score to each transition. And what we found is that even if you look at pretty high transition scores, so this one, we chose a threshold that allows transitioning out of the tissue-resident cells. So with this threshold, so the highest score is 1. Okay, we get a pretty dense graph, which basically allows transitions from the monocytes or from the tissue-resident cells into any of the DMAX clusters. Okay, so in principle, based on this analysis, you can move from the monocytes or from tissue resident cells into the DMAX. Okay, so we wanted to do some additional validation to this hypothesis, this prediction. And we went to our special transcriptomics data. And the idea is that if cluster X come from cluster Y through differentiation, then cells of cluster X should tend to be adjacent to cells from cluster Y more than you expect by chance. Think about this. These cells are related through differentiation. They're not necessarily going to stay together, but they will be close to each other more than you expect by chance. Even if cells from cluster X eventually move away from Y, you still tend to see them in a higher probability close to cluster Y. So we checked this. And what we found is that the edges that connect the monocytes or the tissue resident cells to the DMAX, if you check if they are supported by this adjacency analysis in the special transcriptomics data, the answer is yes. So most of them is what you see here on the left. You can look at pairs of clusters. If it's red, it means that they are significantly co-localized, more than you expect by chance. So there is a support to almost all of these transitions. So everything in red is supported by the special transcriptomic data. The ones in blacks are not. But if you look at them, so there's the transition from this cluster M6 to M11, so early differentiation of monocytes. This edge actually, the threshold I was using was FDR 0.05. This one was 0.06, something like that. So it's very nearly significant. We have these transitions into this specific cluster 17. This cluster was very rare in the special transcriptomic data, so we don't have enough numbers for proper statistics here. But if you look at the p-values, they're in the right direction. So even these edges are supported. So just showing you an example of what this looks like. I'll focus on this cluster M8, which is somewhere in the middle of the differentiation trajectory, either from the monocytes or you can get to it from the tissue resident cells. And based on this trajectory, you expect it to be closer than what you see by what you expect by chance to cells from cluster M13, which kind of goes into M8, and also cells from cluster M10 and M7, which are the next steps in the differentiation. So what you see here, I'm taking cells from cluster M8. Dark blue means the actual probability of being close to any of these other subsets. Light blue is what you expect by chance if there's no preference. And this just reflects the relative frequencies of each cluster. So M5, for example, is very abundant in the special transcriptomics data. So you expect to have a high chance of being enabled to M5. But if you actually look at the actual probability, we don't see this. Now, M5 is not on this trajectory. It's far from M8. So that's kind of what you expect if the trajectory is real. But if you look at the cells that are, the clusters that are close to the trajectory, like M10, M13, M7, then yes, you see that in these cases, the probability of adjacency is higher than what you expect by chance. Basically, it's for M7, for M10, and M13. It's also true for some other classes like M16, but M16 is really very close on the trajectory to M8. And we're not showing a direct transition from it, but it's likely that this is possible as well. Okay, so this is an example of how the special transcriptomic data can be used to lend further support to these hypothesized trajectories. Now, initially, when we were running this, we could not find support for the edges that go from the tissue resident cells, M0 and then M9, into this DMAX. And we're actually missing one edge in each case, an edge from this M0 to M7, the first step of differentiation out of the live 1 minus tissue resident macrophages, and from M5, which is very similar to M9, into M10 and M7. So we're kind of missing these edges. Now, when we looked where these cells are, so we're basically speaking about M7 and M10. In the previous analysis I showed you, they tend to be on the borders of glomeruli or just outside of them. So we hypothesized that this may be the region where the tissue resident cells differentiate. So I mentioned already, the different tissue resident cells are all over the kidney, including the parts that don't have inflammation. But What we hypothesize here is maybe when they're around the glomeruli, this is when they start differentiating into M7 and M10. So we repeated this analysis of co-localization, but limiting our analysis only to the areas around the glomeruli. And when we did this, we found the missing edges. So this is what you see here in green, and edges connecting M5 to M7 and M10, and one connecting M0 to M7. So once you limit the analysis to these areas of around the glomeruli, you see this non-trivial co-localization. So this leads us to hypothesize that the differentiation of tissue-resident macrophages really occurs around the glomeruli, so not in other locations in the kidney. Okay, but we wanted also to get some more direct evidence showing that monocytes can indeed differentiate into DMACs. So we took CD14 monocytes and tried to make them more similar to DMACs. Now, just reminding you that most classes of DMACs have an upregulation of this tissue-related injury program. So we took CD14 monocytes and cultured them in a bunch of different signals that we know are abundant in the kidney. And what we found is that if you culture them for a few days with TGF-beta, they upregulate significantly this program of tissue injury associated genes. If you use interferon gamma on the opposite, in contrast, you actually see a downregulation, significant downregulation of this program. So this kind of gives you a sense of what the signals in the tissue that may be driving this differentiation, although it's quite possible that there are other signals as well. But regardless, it shows us directly that monocytes can indeed become similar to these DMACs. So it's a direct proof that differentiation of the monocytes, at least into DMACs, is possible. Okay, so I'll switch to speak about the last question we had here. I'll try to be quick so we have time for questions. And here we're looking at the role played by the interactions of the immune cells, specifically the myeloid cells, with the T cells in driving disease, specifically interaction of macrophages and parietal epithelial cells. Okay, so parietal epithelial cells are found on the borders of glomeruli. And what it is thought is that these cells get activated, they proliferate, and then migrate to replace damaged podocytes. And this is an essential part of glomerulosclerosis. Now, what we are asking is what is the possible role of immune cells, specifically DMACs, in these processes? So using the same patient groups as before, but now requiring that the early disease have a zero glomerular component. So a zero glomerular CI. So I'm looking at just the glomerular component of the CI and requiring them to be zero. So before any irreversible tissue damage to the glomeruli. So we define this group this way and then compare them to the healthy patients, again, doing this separately for the proliferative and the membranous disease. And what we found is that early on, we have an upregulation of a bunch of pathways that were shown in other work to be associated with the migration of epithelial cells. So there is an upregulation of the programs, and this is in the single cell RNA-seq data. Upregulation of the programs you expect to see leading to PEC migration. Now, we don't see this in pure membranous disease. And recall that in pure membranous disease, there are no DMACs, okay? So that's an important observation. Now, we can use these programs to define a migration score. So we took the genes that are in these pathways and used them to define this composite score. Now, this is something we'll use in the next slides. Now, when you look at proliferative disease and compare them to healthy patients, we see a bunch of receptors that are upregulated. And I'm focusing here on receptors whose ligands are known to drive the activation, proliferation, or migration of epithelial and endothelial cells. So all of these are upregulated, but only in proliferative disease. So this is just showing you where the ligands are expressed on DMACs. So all of them are expressed either by DMACs or differentiated monocytes. And again, as I said, we don't see this upregulation in membranous disease. What we also see in the single cell RNA-seq data is that the expression level of the receptor, showing here on the x-axis in each case, is positively correlated with the migration score. So this kind of suggests that these receptors may have a role in promoting the migration processes. Okay, so we wanted to see if the spatial transcriptomics data validate this prediction. So again, the prediction is that the DMAX may be driving PEX activation, proliferation, and migration through these different ligands that I just mentioned. So the first observation from the spatial transcriptomic data is that PEX do co-localize with different subsets of myeloid cells, so DMAX and differentiated monocytes. And just showing you two examples of the tissue. PEX are shown in green, they are in the periphery of the glomeruli, and you can see that they do co-localize with several subsets of myeloid cells. The second observation is that PEX tend to form aggregates, at least in some glomes. And you see these darker areas, areas where you have higher density of PEX. Now, if you look at these aggregates, they have a higher migration score. So this tells us that the migration score is indeed meaningful in reflecting tissue pathology. They also have a lower interferon score. Now, going back to the single-cell RNA-seq data, when the chronicity increases within the glomes, we actually see a decrease of the interferon response. So this is consistent with this observation. And again, suggesting that these aggregates of higher density of PEX are associated with irreversible tissue damage. Now, we then looked at PEX that are not found in aggregates, so still in low density areas, and we asked how the migration scores changes if they are neighbors of different myeloid cells. And what we found is that there are several subsets of DMAX that if they are neighbors of these PEX still in low density areas, they are associated with an upregulation of this migration score in these PEX. Okay, so again, this is consistent with the idea that these DMAX induce these changes in the PEX even without the formation, before the formation of these aggregates. If you look at the specific expression of 3 of the ligands that we had, which were in the panel of the xenium data, we did see a significant, though not very dramatic, increase in the migration score if these are expressed by monocytes and macrophages around the PEX. But given these small changes, this suggests that there are probably other ligands or maybe a combination of ligands that is important to these shifts. Still, they are statistically significant, especially when you compare them to cases where they are not expressed, where you don't see any shift, any increase in the migration score. Okay, so I'll quickly summarize, and we have like 10 minutes for questions. What we get from this work is a pretty detailed catalog of all immune cells in the kidneys. I'm focused on myeloid cells, but we have similar catalogs for other cells. I showed you that the frequency of these cells is related to the ISM class in AI and CI. Specifically, early disease, the low CI, low chronicity, is associated with a response that's more dominant by so centered on monocytes, macrophages, and CTLs. And later in disease, we see an increase in the frequencies of CD4 T cells and B cells. We also see within the myeloid compartment an expansion of dendritic cells as the chronicity increases. And only in proliferative disease, we see an expansion of these DMAX early in disease, but then they contract later in disease when the chronicity increases. In terms of transcriptional programs, early in disease, you see the interferon score going up, but then it goes down. And in true membranous disease, it basically goes down to normal levels. And later stage of disease, we see an upregulation of the machinery associated with antigen presentation and processing, consistent with this shift of a CD4-based response. And in just proliferative disease, early we have upregulation of phagocytosis associated genes, but then they go down later in disease. I mentioned the idea of convergent differentiation, where both monocytes and resident macrophages adopt similar states. And what the spatial transcriptomics data tells us is that the monocyte differentiation mostly occurs within the glomes, and the arm differentiation occurs around them. And I gave you some evidence that DMAX and differentiated monocytes promote glomerulosclerosis. And again, an important point in this analysis was that we can never have an experiment where we remove DMAX from human patients and see that there's no effect on the PEX, right? But nature kind of did this for us in pure membranous disease. So this is an example where DMARCs are absent and the PECs don't change the state. So that's probably the closest we can get to any point perturbation in immune patients. Okay, so I'll stop here and I'll be happy to answer any questions you may have.
Thank you very much, Arnon. This is an excellent talk, and we've had a very high attendance, I think, reflecting the interest and the excitement that people have about these results. And so for the audience, if you have questions, please type them into the Q&A section. Jessica, I think, do I have access to that? Because it says only the host and co-host will see them.
Questions should go into the chat. We don't have a Q&A
Oh, we don't have.
forum. So chat is where all questions will go. And then everybody, if you make those public, everyone should be able to see your question.
Okay, sounds good. So let's do chat. This tells you that when one person who used to manage the Zoom is not here today.
The change is that we had a webinar feature on the other account, and those are no longer existing in the newer Zoom account, so we don't have webinar feature, which is what creates the difference.
Mm-hmm.
I'm not seeing any questions yet.
Yes, I can start with some questions. Arnon, are there any hint source of the interference from this data?
So can you repeat the question? It was
are there any hints used from this data about the sources of the interference?
Yeah, so that's a separate question. We kind of started analyzing this. There is an obstacle here, which is the high overlap between the signatures of type 1 and type 2 interference. And type 1 interference affects thousands of genes, kind of depending on how you define this. But if you look at something like the interferon data set, it contains thousands of genes. You can limit this to smaller sets of genes, but it seems to be dependent on the cell type that is exposed to the interference. So what you need to do is actually construct a resource that tells you what the interferon response looks like over time if you give type 1, type 2, different doses, a combination for different cell types. And we are in the process of constructing this. But the data that we already have suggests, to me at least, that it's probably a combination of type 1 and type 2 effects. There's plenty of interferon gamma in the tissue, mostly from T cells and NK cells. And we do see a correlation between where the cells are and the effect of the interferon on the cell. So if you I kind of ran through this, but not all subsets of myeloid cells have the same difference, the same changes in interferon response. And we think that's probably because they have different proximities to T cells and NK cells as disease progresses. So what these data suggest to us so far is that these are the local effects. Not all cell types are affected the same way. So it's not just a systemic downregulation, although that's probably happening as well. It depends on both type 1 and type 2 interference. And it's probably a combination. So there's a paper a few years ago, I think like 2 years ago or 1 year ago, that came out that showed that the signature of type 2 interference is really local. It's like 30 microns. So this by itself is not explaining a general decrease, but there could be like an amplification where you have less CTL, so less interferon gamma, less cell death, so less type 1 interference as well. And type 1 interference propagate in the tissue, right? Because cells, any cells, they can sense it and respond, right? So that's probably a combination of these two effects. But I think it's a very complex process. The general thing, though, is that it goes down with chronicity. That one is pretty robust. And by the way, we see this in urine. And it turns out to be a very strong indication of the chronicity. If you just follow the interferon response in urine over time, this seems to be a very good marker.
Okay, thank you. And I think we have a question from Sean Jackson.
Yeah, I was typing and then I was getting confused about what I was trying to say. For the final part, where you had interactions between the diseased macrophages and the PECs, you've already shown that the diseased macs go into the gloms. And so in some sense, the fact that the parietal epithelial cells always surround the glom, you're always going to have some spatial interaction. So my question is the spatial interaction more than spatial interactions with podocytes, mesangial cells, etc. Like, how do you distinguish? Because you're making an argument that these cells are specifically interacting with one type of cell, which is spatially localized within the glom. How do you avoid the circular argument for you?
So first of all, we're focusing on, I'm not saying that they're the only one they interact with, and I'm pretty sure that there are feedback loops within them. We actually have similar analysis with glomerular endothelial cells.
Yes.
And you do see some correlations there as well, not necessarily with the cells, exactly the same cell types. And it is related also to T cells. So what I'm thinking is happening there is a much more complex process where all of these cells communicate. There's also signals that go from the plex to the myeloid cells. I kind of skip that, but we see this. We see these correlations as well. So it's a complex process where you have, all of this is happening there. I'm not going to say that we can completely separate these processes, and I don't think they can be separated. I think they are highly dependent on each other. But I think an interesting observation is that where you don't have these DMARCs, you don't see these changes. And by the way, you see this also within a specific sample. So not all gloms have DMARCs in them. And where you don't have the DMARCs, you don't see these changes. So that, to me, is a more direct evidence of this relation. But it's a complex process for sure. The claim that we're making is that DMARCs are driving it. So that without DMARCs, it's not happening or happening less, let's say.
Yep.
And yeah, I guess we have a question from Sheep. Are there any neutrophils found in lupus nephritis kidney by single cell or spatial transcriptomics? If so, what's the percentage of neutrophils found?
Neutrophils are extremely tricky. They don't survive freezing and thawing, so we don't see them in the dissociated kidney. Or that's the other issue, they're not profiled well by 10x. So even if you have neutrophils in a sample and load into 10x, human neutrophils are very difficult to see. We are using there's a similar tool to 10x that does better looking at neutrophils, and we are looking at them in urine. They are there. So it's kind of difficult to say how they got there, if they're from the kidney or from, let's say, other parts of the urinary tract. But they should be there. Now, in special transcriptomics, again, it's not easy to capture them. And a lot of it is technical, I think. So I'm pretty sure neutrophils are there. I'm pretty sure they're doing something. But they're extremely difficult for us to study them.
And I was wondering, Arnon, you mentioned having follow-up biopsies, but I'm not sure for this part of the study, did you have treatment response data, and was there any correlation with certain cells?
So that certainly complicates things. Now, the study was not designed as a clinical trial, so we have a whole bunch of different treatments. We're taking them as a covariate, but I don't think you'll ever get a full clean avoidance of this effect. I can tell you that we did look at some clinical trials data where we have repeat biopsies, not single cell data, but bulk RNA-seq. And we do see some changes there that are consistent with this interpretation. This is not what I mentioned, though. We do have some samples where we have repeat biopsies that are now analyzed the same way I showed today. So we have the data, we're doing the analysis, we're just missing some clinical features. We don't have the chronicity, for example, there yet, so we're trying to get there. So yeah, I mean, look, in real world, you can never completely avoid the effect of treatment, right? These patients are going to be treated. You would need a large sample to be able to completely explain this, like hundreds of patients, I think. So this is not this study. There are attempts by us and other people to get repeat biopsies in large numbers and try to think about this. I think it's tricky. It's always going to be tricky.
And do you have, doesn't have to be a repeat biopsy, but for those patients that you have the biopsy for, do you have, like, I don't know, 6 months later or a year later, did they have a good response? No.
So we do have the response data, and it does seem to change. So if I limit the analysis just to the non-responders, it looks different than responders, yes. The numbers are not great. We have 11 repeat biopsies, and about half of these are responders. So yeah. you can use this for validation, not for inference.
Yes.
All right, well, we are at 10 o'clock, and I want to make sure that everybody can get on to the next thing that they have. I want to thank everyone for joining us today. And for our speaker, we really appreciate you joining us. If you have additional questions, I think some of you are meeting with him throughout the day and can bring those up at that time. Otherwise, thank you and have a great rest of your day.
Also, feel free to email me questions if you have any.
Great, thank you.
Thank you.